everybody. Thank you all for being here. Today, we're going to be taking a look into my research on the fungal fermentation of brewery spent grain. So, to provide some context and background to my research, it's first important to understand the significance of the larger issue of food waste generally, the more specific context of brewery spent grain, or BSG, and how resultant risks from these issues can be mitigated through fungal fermentation. Now, just over 1 billion tons of food is wasted per year, according to the United Nations Framework Convention on Climate Change. Furthermore, 8 to 10% of global greenhouse gas emissions annually are resultant from this food waste. 13% of this food waste also occurs during the supply chain, which can include food waste from the manufacturing process. Now, one context of food waste during the manufacturing process comes in the form of brewery spent grain, or BSG. Now, BSG is the single most abundant waste byproduct of the brewing industry, and it's composed primarily of malted barley, which has been semi-fermented over the course of the brewing process. Now, just over around 36.4 million tons of BSG is generated per year, according to Nyhan and co-authors. Furthermore, 70% of this BSG is subsequently repurposed and used as an animal feed, primarily for cattle, according to Tarif's 2022 study. Now, when high fiber feeds such as BSG are fed to cattle, this has been shown to increase methane emissions from cattle, according to Santander and co-authors. This presents a clear need for a method by which we as humans can increase BSG's digestibility, thus mitigating the risk of increased methane emissions when fed to cattle, representing a large use avenue of this waste byproduct. Luckily, fungal valorization, or increasing of value of BSG, has been shown to enhance its digestibility by not only degrading fiber, but also enriching protein concentration, mitigating, thus, the risk of increased methane emissions. So, for my review of the literature, the first study which I encountered, and possibly one of the most formative studies on my understanding of the topic generally, was Canedo and co-authors' 2016 study in Brazil. And so, in this case, researchers looked at the fungus Rhizopus oligosporus, which is a very commonly researched fungus in the literature. And for a substrate, they used barley BSG, which is traditional practice for brewers, as previously stated. Researchers also controlled for 2 treatment groups, these being nitrogen source and initial moisture of the substrate. And finally, in this case, fungal suitability for BSG valorization was quantified through soluble protein enrichment, or the increase in soluble protein concentration from substrate to fermented product. Now, Ogunjobi and co-authors conducted a very similar study to Canedo's. However, in this case, they used Aspergillus oryzae as opposed to Rhizopus oligosporus. Now, A. oryzae is another very commonly researched fungus in the literature. And again, Ogunjobi and co-authors used BSG as a substrate for their fungal growth. However, this BSG was composed of sorghum due to the fact that researchers were located in Nigeria, in which the importation of malted barley is illegal. So, researchers in this case did not control for any different treatment groups specifically. However, again, to quantify fungal suitability for valorization of BSG, they once again used a measure of protein, in this case, crude protein concentration as opposed to soluble protein enrichment. Now, the only study that I was able to find in the literature that directly compared Rhizopus oligosporus and Aspergillus oryzae was Mitchell and co-authors' 1988 study. Now, in this case, they did not use a BSG substrate for experimentation, instead using cassava starch. Furthermore, they controlled for a plethora of variables, including pH, starch, glucose, urea, and salt concentration. Now, the dependent variable, or the method by which they quantified fungal valorization suitability of the substrate, was not a measure of protein like Kanedo and Ogunjobi studied, but instead colony radial growth rate. So, I was able to identify a gap in the existing scientific literature, being that researchers yet to determine which fungus, Rhizopus sporus or Aspergillus oryzae, is more effective in the protein enrichment of brewery spent grain through solid state fermentation, or SSF, also known as fungal growth. Now, this led to the development of my research question, being under identical and controlled growing conditions, which fungus is more effective in increasing the soluble protein content of BSG. Now, the methods which I developed in order to address this research question involved first preparing and growing samples, and then extracting a protein liquid isolate, which was later measured for protein concentration. So first, BSG was collected from a local brewery, at which point it was divided into 3 different treatment groups, one for each fungus, as well as one uninoculated controlled treatment. Then 4 40-gram samples of each of these treatments were created and grown in an incubator set to 30 degrees Celsius for 3 days. After incubation and growth, samples were processed to extract the supernatant, or liquid protein isolate. And this was done by dissolving protein into a phosphate buffer, which required blending 10 grams of sample with 90 milliliters of phosphate buffer for 1 minute, and then stirring this solution using a magnetic stir bar and stir plate for another 30 minutes to fully dissolve all soluble protein in the solid BSG. Then, this protein liquid had to be isolated from solid BSG sample residue, which was done by centrifuging at 15,000 RPM for 15 minutes. At this point, the protein liquid, or supernatant, could simply be decanted from the solid residue condensed at the bottom of Falcon tubes to later be used to quantify protein enrichment. Now, the method by which I quantified soluble protein concentration was the Bradford assay from 1976. This is a very commonly used method in the literature, used by many studies looking at solid state fermentation and BSG enrichment, including Lee and Wang, Mora Lugo and co-authors, and Kumaran and co-authors. So the Bradford assay involves first adding a reagent or dye to the protein sample. And when in presence of protein, Bradford reagent bonds with N-terminal amino acids, becoming blue. Now, this resultantly causes this blue solution to absorb orange light in the 595 nanometer range. This absorbance can be analyzed in a spectrophotometer, which yields an absorbance value, which essentially quantifies how blue the sample is. Now, on its own, this absorbance value doesn't really mean much of anything. And in order to calculate protein content based on this absorbance value, we need to use a standard curve. So this is created by making protein solutions of varying known concentrations. Usually this is 0, 10, 20, 40, 80, and 100% protein. The standard protein, which is most commonly used in the Bradford assay, is bovine serum albumin, or BSA. And then these protein solutions are then mixed with Bradford reagent once again, which will become more blue in higher concentrations of protein. Then these solutions are analyzed in a spectrophotometer, yielding an absorbance value, which can be plotted over each solution's concentration, creating a linear regression model here. Now, this is my standard curve. And using this regression, we can, when given a sample experimental absorbance value, such as 0.6 here, calculate the corresponding protein concentration. In this case, just about 0.87 milligrams per milliliter. Now, for my results, right out of the incubator, multiple disparities between sample conditions were immediately visible. Notably, Rhizopus sporus samples 1 and 4 were the only samples to demonstrate visible mycelia growth. Furthermore, control samples 1 and 3 were significantly more moist than any other experimental samples. Now, after samples were processed and protein was quantified, it was seen that the single sample with the highest soluble protein concentration was control sample 3. As you can see here, control 3 was one of the moist control samples, as indicated by the blue cell shading, and it had a soluble protein concentration of just about 0.3 milligrams per milliliter, making it the single most proteinaceous sample in my experimentation. Now, additionally, it could be seen that moisture, higher moisture specifically in control samples, was correlated with increased protein concentration. As we can see here, control samples 1 and 3, which were moist control samples, had protein concentrations of 0.2 and above, while the drier control samples, 2 and 4, had protein concentrations just around 0.1, almost half of the soluble protein concentration of moist control samples, representing a clear correlation and disparity. Here we have a plot of each sample's soluble protein concentration grouped by treatment group with some descriptive stats. And as we can see, Aspergillus oryzae samples had the highest soluble protein concentration on average, followed by Rhizopus samples, and then control. However, control samples had far and away the highest standard deviation of any treatment group due to, again, that disparity in soluble protein concentration, which was correlated with moisture. Now, after statistical analysis was conducted, which involved t-tests conducted between fungal treatments, as well as between each fungal treatment and control, as well as an ANOVA test, which was conducted between all 3 treatment groups, the resulting p-value for each of these tests did not convey any convincing statistical or statistically significant difference between soluble protein concentrations between any treatment group. However, when control samples 1 and 3 were considered outliers due to methodological error and removed from the data set due to that methodological error in moisture control, then when statistical tests are rerun, a t-test between Rhizopus and the control, as well as Aspergillus oryzae and the control, both yield p-values more than low enough to demonstrate considerable, statistically significant, higher protein concentrations in fungal fermented samples than control samples. Now, for my discussion and conclusion, first and foremost, this is the first side-by-side comparative analysis of these fungi on a BSG substrate. Thus, my findings can't be compared with anything else in the literature. And furthermore, my results indicate, however hesitantly, that due to the lack of statistically significant difference in protein, in soluble protein concentration between fungi, that they may be equally effective in the protein enrichment of BSG. Finally, the fungi showed a considerable disparity in mycelia growth volume, with Rhizopus samples being the only samples to demonstrate visible growth once again. However, they did not display this disparity in protein concentration, which could indicate that Aspergillus oryzae samples enrich protein more effectively per unit of mycelia volume. Now, my study had the following limitations. First, commercial starters were used in this sample as inoculants, used in this study as inoculants, sorry. Now, these contained different ingredients between each of them, as well as different spore concentrations and spore densities. Thus, results in protein yields between fungi could not be directly comparable. Additionally, my sample size was too small to determine results with any high level of confidence, using only 4 samples for each treatment. BSG is an inherently non-sterile substrate. Thus, existing biological activity in my BSG could have resulted in unintended protein enrichment of control samples during incubation. And finally, experimental samples exhibited significantly lower growth than that found in a pilot test which I conducted prior to experimentation using household materials. Thus, one could hypothesize that had these fungi been presented to optimal growth conditions, that results might not be the same. Now, finally, potential avenues for future research should consider A, using a larger sample size to determine results with high confidence. Additionally, controlling for initial moisture of the substrate, using pure spore inoculants or inoculants of equal spore density, and quantifying protein both before and after incubation and growth to accurately quantify protein enrichment from substrate to fermented product. Thank you all. Here are my references.
Thank you, Liam. Give me a minute and I'll grab a question for you here.
Of course.
How did you determine which results generated by your research method were most important in informing your new understanding?
Can you say that once more?
Absolutely. How did you determine which results generated by your research method were most important in informing your new understanding?
Yeah. So first, I would like to say that my results weren't necessarily very conclusive due to the small sample size, lack of statistically significant differences, not only between fungal groups, but also between fermented treatments and control groups. My results can't be expressed with any high level of confidence. However, I think that the most formative result for my new understanding was not about fungal performance, but about the efficacy of my methodology, which I was able to identify multiple limitations, including small sample size, commercial starters, and more. And I think that my new understanding is primarily based around these limitations and how future research could fix these limitations by using a larger sample size, pure spore inoculants, and other things such as quantifying protein before and after growth to yield a more accurate new understanding.
Great, thank you.
Well done, Liam.
Thank you.
What are the real-world implications or consequences related to your findings?
Yeah, so I set out with the research goal of determining which fungus, Rhizopus or Aspergillosaurus, is more effective in the protein enrichment of BSG, which is a measure or a method by which we can quantify how effective these fungi are at valorizing BSG or increasing its digestibility. Now, when BSG is made more digestible, this can mitigate the risk of increased methane emissions from use as a cattle feed, which is one of the primary use outlets of BSG. Now, if we can figure out how to most effectively valorize BSG, this could inform future practice in the industry to create a more sustainable method of use of this waste byproduct.
Again, well done, Liam. We're
Thank you.
impressed. So I'm going to ask you to think back to the initial curiosity that sparked your inquiry. What other curiosities do you have, and how has this process prepared you to explore them?
Yeah, so my inquiry was primarily sparked by simply looking at some science news articles. I stumbled across one which was detailing the valorization of human food waste using a different fungus, which was then repurposed and used in fancy restaurants. And I now, before coming to this project, was looking at a linguistics project, thinking about how baby talk develops, as well as this, of course, fungal fermentation research. And my experience with this project has taught me how to take an inquiry like this and research existing scientific literature, immerse myself in it and understand it, and then use this to develop a methodology and defend a finding based on a gap in the literature.
Can I just go back to what curiosities do you have?
Absolutely.
How did this promote some curiosities in you?
Yeah, so relative, again, I said I still am interested in linguistics to a pretty high degree. That was a big consideration for this AP research project. But also after finishing this project, I have had some new ideas as to how this research could be carried forward, or new curiosities. Those being primarily focused around how other food wastes can be repurposed, not only to mitigate emissions and promote sustainability in their discard, in like the process of being discarded, but also to be reused and recycled and consumed again.